Today we are going to continue our muscle physiology. Okay? Up till now, we've talked about all of this, kind of an overview and brief tidbits. And last class, we went over what happened and how we generated an action potential, right? Release of acetylcholine from the neuron, crosses the synapse, binds to its receptor on the motor end plate. Ions move in and out, okay? Stimulates. What ions move into the muscle cell? What ions move in? Chris? Okay, into the muscle cell. Calcium would move into the axon to stimulate release of acetylcholine. Okay? Then acetylcholine crosses the synapse, right, and binds to its receptor. Now what ions, that channel's open, what ions move into the muscle cell from there? What ions move in? Sodium, right? Sodium moves in, and what ions move out? If sodium moves in, what one goes out? Potassium, okay? And that causes a change in the polarity. What do we call that change in polarity? What's the word for that change in polarity? The change in the electrical potential, the electrical difference across the inside of the outside of the membrane. What is the word that when we change that, what is that called? Depolarization. Okay? And then if we have enough of a local depolarization or an end plate polarization, that can cause other channels nearby to open. What channels would open? If we reach a certain amount difference. And what's that certain amount difference called? There's a certain point that we have to get to. Not equilibrium, close. Threshold. We have to reach that certain difference. Once we reach threshold, and then now we generate an action potential. And what channels are stimulated to open first? Sodium channels. Sodium channels open, sodium rushes in. And then that's a big change now, right? The graph we showed goes really high, and we have depolarization that happens. And we go all of the change in potential goes all the way up to positive 30. And then sodium channels close, and what channels open? Potassium channels open. And then what's that process called when potassium channels are open? Repolarization. Okay, and then potassium channels are open. Potassium rushes in. And then eventually potassium channels close after repolarization. We get back to resting. And then what happens with where sodium and potassium were? They were flipped in their original position, so they get pumped back out, right, by the sodium-potassium pump. Okay? Now, all of that action potential that is generated from the motor end plate or the neuromuscular junction and then travels down the sarcolemma, right, in a stepwise progression. Each little area of the membrane stimulates the next little area, and it continues in a one-way direction, right, down the sarcolemma, and eventually that action potential is transmitted into the T tubules. So what we're going to talk about first today is excitation, action potential, and how it's connected to the events of contraction. And how does contraction happen? What's the model that we use for contraction? What's it called? Remember, we said last week, we talked about the sliding filament model for contraction, where actin and myosin filaments are going to slide across, right, parallel to one another. So somehow there has to be a way of connecting the events that are happening on the membrane of the cell to the mechanical events that are going to happen on the interior of the cell. And that's what excitation-contraction coupling is. Okay? So, action potential goes down the sarcolemma. It's pretty quick, right? It actually ends before contraction ends, or the action potential actually completes before contraction even starts, okay? But the connection or the result of an action potential is that it causes a rise in intracellular calcium, which then leads to contraction. And where does that intracellular calcium come from? Where is it released from? What part of the cell stores and releases calcium? The sarcoplasmic reticulum. Okay? So if the sarcoplasmic reticulum is where it stores and releases calcium, we have to have a way of connecting the excitation, the action potential that's happening on the membrane to somehow connecting it to the sarcoplasmic reticulum, and that is where the triads come in. Okay? The latent period of a muscle contraction is the time when this excitation-contraction coupling occurs. There is a time point. Okay? It takes a little while to go from action potential runs down, open calcium channels, and then calcium rushes into the cell, and then all the events of contraction occur. Okay? There's a short period of time. That is called the latent period. During that time, another action potential can't come down the sarcolemma and do anything more, right? The muscle in this case is kind of non-responsive, okay? So the latent period is the time between an action potential or a stimulus and contraction beginning, okay? It is equivalent to the time it takes to relay the message from the membrane and get enough calcium released in the cell to stimulate contraction. Okay? Latent period. So, action potential propagated down the sarcolemma and through the T-tubules. All right? And in the T-tubules, we have voltage-sensitive proteins that stimulate calcium release from the sarcoplasmic reticulum. These are basically equivalent to, voltage-gated calcium channels. Okay? And calcium necessary for contraction to occur, because calcium is going to bind what protein? What protein have we talked about in muscle within the filaments that binds calcium? What protein binds calcium related to muscles? Here, I'll give you a start. Tropomyosin? Does tropomyosin bind calcium? Tropomyosin is attached to the protein that binds calcium. So what's the protein that binds calcium? Troponin. Okay? Troponin. Okay, troponin. Okay, troponin is going to bind calcium. Okay? So here's excitation-contraction coupling in one picture. Okay? Action potential is generated in the motor end plate. All right, you get a local depolarization, end plate polarization, and then that's transferred to the sarcolemma and generates an action potential. Action potential propagated down the sarcolemma, down the T-tubules. And as it goes down the T-tubules, it stimulates the channels that are present in the terminal cisternae of the triad. Remember, the terminal cisternae are the end points, parts of the sarcoplasmic reticulum. So as that action potential runs down the T-tubule, it causes these calcium channels on the inside of the cell to open. When those calcium channels open, calcium is released into the interior of the muscle. Okay? That's the link between the events that happen at the membrane to what happens inside the cell. Okay? So then, calcium, okay, here's same thing that's happening right here. We just talked about action potential comes down. Okay? It causes release of calcium into the interior of the cell. Now, calcium binds to troponin. When calcium binds to troponin, troponin changes shape. And the role of troponin is to regulate where tropomyosin is. Tropomyosin, in the absence of calcium, is covering these actin binding sites on, or the myosin binding sites on actin. Okay, so now myosin can't bind at this point. But when calcium is available at there's enough of it, it binds to troponin, troponin changes shape, and moves tropomyosin out of the way. Okay, so here, tropomyosin has been moved because calcium bound to troponin. Now it exposes all of these binding sites on actin, myosin binds actin, and then it bends. And that is the start of contraction. Okay? So the release of calcium, stimulating the release of calcium, is how we link action potential to contraction. Okay? That is excitation-contraction coupling. Right? Is just controlling and stimulating the release of calcium in the muscle. Make sense? I'm seeing nods. It's good. Most people are kind of like painted, are good. Okay, following along. It's good. Okay, so here's we can just go this in kind of stepwise production. Actin, tropomyosin, troponin, no binding because tropomyosin is covering the binding sites for myosin on actin. Okay? So now calcium is released in response to an action potential coming down the T-tubule and causing and opening those voltage-gated channels. So if calcium flows out, what's going to happen? Calcium here is going to bind to troponin. All right? Calcium binds to troponin. It binds, troponin changes shape, and when it changes shape, it moves tropomyosin out of the way. And now those active sites for myosin are exposed. Okay? Myosin binds, and contraction begins. Okay? Myosin binding to actin, what is that called? When myosin binds to actin, you're forming what? What did we say that was? Crossbridge, thank you. I didn't hear it very well. These masks can't hear, I swear. Crossbridge, you get formation of a crossbridge. Okay? And when the crossbridge forms, myosin bends and contraction begins. Okay? That's the whole excitation-contraction coupling process. Okay? Questions? And on excitation-contraction coupling. Now, when the action potential is gone, the stimulus is away, calcium will get pumped back into the sarcoplasmic reticulum. The levels of calcium go down. Now there's not enough to bind to troponin. All right, and then, so calcium is no longer bound to troponin. Troponin goes back and changes shape back and moves tropomyosin, covering the active sites. Okay? So, altogether, the channels that are involved, okay? Couple different ones. Nerve impulse reaches in the axon terminus. We have voltage-gated calcium channels, okay? Then we have when that stimulates the release of acetylcholine from the vesicles. Acetylcholine goes across the synapse and binds to ligand-gated channels, right, in the motor end plate. And then after that, a local end plate potential stimulates the opening of voltage-gated sodium channels. Okay, and you get an action potential propagation. And then from there, what else is not written on here? What channel is not included in this? And I left it out. Which one during the propagation of the action potential, voltage-gated sodium channels, what else are we missing? All right, we also need to include our voltage-gated potassium channels. And that's during repolarization. Okay, and then finally we have our voltage-sensitive proteins, voltage-gated channels, right, which stimulate the release of calcium from the sarcoplasmic reticulum. Okay? So we're linking the change in the voltage potential in the membrane to controlling the release and storage of calcium, which is necessary for contraction. Right? You could consider the calcium at a certain concentration to be the switch on where the contraction can occur or not. Okay? And so our role of calcium then is that it binds to troponin. Okay? And so at low intracellular calcium, there's very low levels. Calcium in this case is not being released from the sarcoplasmic reticulum. In this case, tropomyosin blocks the sites on actin. The myosin heads therefore cannot bind, and the muscle fiber is relaxed. Now, give me 3 bullets or what's going to happen once there's calcium's being released. What's first? So calcium's going to bind to troponin. And what is that going to do for tropomyosin? It's going to get out of the way, right? It's going to move the blockage, remove the block, thereby exposing the actin sites, and the muscle then can contract. Okay? So calcium binds to troponin, changes shape. So calcium binds to troponin, shape change, and it moves tropomyosin away from those binding sites. Once there, the affinity of myosin for actin is very high. It likes to bind, and as soon as it binds, it bends, causing the sarcomere to shorten and overall muscle contraction. Okay? When the stimulation is stopped, calcium is pumped back into the sarcoplasmic reticulum and contraction will end. Right? Not enough calcium is going to be present to bind to troponin. Troponin goes back to its normal shape, and as long as those cross bridges are broken, tropomyosin will recover the binding sites, and therefore the muscle will be relaxed to prevent binding. Okay? Questions? No. Okay, great. So, let's talk about the Crossbridge cycle. So as long as those sites on actin are exposed, myosin will bind, okay? And then it will go through this cycle of forming and breaking and finding another spot on the filament to bind to, right? So it will bind, it will break, it will bind, it will break. But really there's 4 steps in this cycle, okay? And as long as the stimulation is there and calcium is present, okay, and enough ATP, this cycle will continue, okay? So this is called the Crossbridge cycle, 4 steps of binding, bending, breaking, and to keep the alliteration bouncing. It bounces to the next available site. Okay? So, crossbridge formation. What is a crossbridge formation? This is myosin binding to actin. That is the formation of a crossbridge. Okay? So in this case, in order for binding to occur, myosin is in a, how we'll call a high energy form. It is like sprung. It is pulled back and there's like a spring. Okay, you can imagine it, I use the analogy of a mouse trap, right? It is in a high energy, ready to spring position, okay? And as soon as it binds to actin, that spring is sprung and contraction occurs, all right? That bending, right, is what we call a power stroke, okay? The myosin head here pivots and pulls the filaments into the end line. That is bending. Okay, so crossbridge formation, binding of actin to myosin happens, and as soon as the binding happens, the myosin head, because of its what we call a cocked position, right, it bends right away. Power stroke. Okay? From there, now the next step is going to be, we've done binding, we've done bending, the next one is breaking. In order to continue contraction, we have to break the association of actin and myosin. Okay? And this is important. This is where ATP is used in contraction. Okay? ATP is used, in this case, to break the association and reset myosin back into its cocked position. Get it? Okay? So, breaking is where ATP is hydrolyzed and myosin is reset. Okay? So, the cocking of the myosin head there, okay, occurs. Right? And that's during hydrolysis and breakdown of ATP and puts it in a high energy state. The high energy state means it's ready to bind and bend right quickly. Okay? And then from there, once the crossbridge association is broken and myosin is reset, myosin can seek out another actin binding site in order to do the whole cycle over again. And this again will continue as the sarcomeres get pulled closer and closer together, as long as calcium is present and as long as the stimulation from the nervous system is present. Okay? So you will continually run through this particular cycle, right? Crossbridge formation or binding, right? That leads to the power stroke that happens, bending. We have to then disassociate myosin from actin, utilizing ATP, okay? That's the breaking. And then it bounces to the next available binding site to continue the process as long as the stimulation and calcium are available. Make sense? So let me ask you, what happens when a muscle runs out of ATP? What part of the cycle is disrupted? What particular part? Someone said it over here. The release, the breaking, right? So in this case, if we run out of ATP, a muscle runs out of ATP, myosin is going to stay bound to actin, okay? And the muscle is going to stay in a contracted position, okay? And you may have heard that in what we call rigor mortis, okay? Rigor mortis is what happens to muscles in the body after death, okay? So your crossbridge detachment or breaking requires ATP, okay? After about 3 to 4 hours after death, right, your muscles begin to stiffen, right, with weak rigidity at 12 hours post-mortem, okay? Why do the muscles begin to stiffen? Well, because dying cells, they bring in calcium. It's equivalent to releasing calcium from the sarcoplasmic reticulum. Calcium comes in, the levels of calcium go up in the cells. That means troponin, calcium binds to troponin, tropomyosin gets out of the way, and now those binding sites for myosin are exposed. What do? Myosin binds to actin, and it bends. But now there's no available ATP that's present in cells to break the detachment. So the muscles stay contracted. So the body, quote unquote, a stiff, right, is in a contracted position for a period of time. Okay? So there's no ATP that's generated to break that dissociation. Eventually the stiffening wears off when calcium in the cells break down after a period of time. Okay? But you can time this, you can estimate time of death based on whether or not the body is stiff or not. Usually if it's in a state of rigor mortis, it's less than 12 hours that the body has death after the time of death. Okay? Questions? Okay, so that brings us to, where do muscles get ATP for energy for contraction? Okay? There's a couple different ways. We've already discussed, I mentioned that the muscles have myoglobin, which is an oxygen-binding molecule, right? It has glycogen, so it can make and store glucose. So it has all the stuff necessary to do cellular respiration, including mitochondria, to produce ATP, okay? But there's already some ATP that's already set up in your muscles, okay? So your muscles have a little bit already in storage form, okay? They have enough for about 5 seconds of intense exertion. Done. That's all my biceps have, okay? For those skeletal muscle cells that I just recruited to flex my elbow, they're out of ATP, okay? So now the question then is, can you flex a muscle more than 5 seconds? God, I hope so. Yes, right? So there are a couple different ways. Do you exercise for more than 5 seconds? I hope so. There are several different ways that we have to be able to generate ATP. We have immediate, short-term, and longer-term processes to generate and produce ATP for muscles to contract, okay? The first quick one is what we call phosphate transfer, okay? So in this case, phosphate is going to be transferred from one molecule to ADP to make ATP. We're just moving one phosphate from one area to another to make a quick amount of energy, right? To just, hey, we need ATP right away. We don't have time for anything else. Boom, 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 move it and shuffle, okay? That's one. Second one is short term, happens pretty fast. That's through the process of glycolysis. That's anaerobic, okay? It happens very quickly. And then finally, long-term supply is cellular respiration. Use of oxygen, breakdown of glucose. That takes a little bit more time because it has to go through the electron transport chain. Oxidative phosphorylation produces a bunch of ATP, okay? So those three ways. Now, we've already discussed two of them, right? When we did cellular metabolism, we went over glycolysis and we went over cellular respiration. We're going to visit them a little bit in terms of what it means in terms of muscle. Let's talk about phosphate transfer, okay? Phosphate transfer is the movement of one phosphate group off of a molecule and attaching it to an ADP, adenosine diphosphate to make ATP, okay? So a couple different enzymes that do this. Number one is what we call myokinase. Kinase, anytime you see kinase enzyme, it means it's going to move or add a phosphate group to it, to a molecule, okay? So a kinase enzyme, like a hexokinase, it's going to add a phosphate, okay? Simple as that. Kinase adds a phosphate. So in this case, myokinase takes one phosphate off of ADP and puts it on another ADP, right? Each of these have two phosphates. 2 plus 2 is 4. We're going to make 1B3 high energy adenosine triphosphate. That's a 3, not a 2. And the other molecule is going to have 1. 3 plus 1 is still 4. Okay? We're just shuffling so we can use the ATP for other stuff. Make sense? Okay. The other enzyme is called creatine kinase. All right? Creatine kinase. And creatine is a molecule that's in your muscles, and it is normally in a phosphorylated form. It has a phosphate on it. Okay? So what ended up happening is you take the phosphate off of creatine phosphate and put it onto adenine triphosphate. Have any of you heard of a supplement called creatine? Yes. Okay? The basis of this supplement, your muscles have some creatine in it. Okay? And the creatine that you produce from your absorbed from your diet is put in the muscles, and it's phosphorylated, and it's there for this immediate transfer. Okay? But if I take more creatine from the diet, I can increase this amount. Right? Here's the science behind it. If I increase the amount of creatine that's in my muscles, it's automatically going to be phosphorylated. And when I start to work out, I have more available creatine phosphate, which then can lead to more ATP. And let me, when I'm bench pressing, a couple extra 2-3 reps of working out. Right? And that's true. Okay? The supplement that people take, creatine powder, you take probably 10 times more in excess than you'll ever need. So when you take it, the little warning label says, drink or plenty of water with this. Okay? Because your kidneys filter out the excess. Right? And one of the markers of kidney function is an enzyme or byproduct called creatinine. And it's a breakdown of creatine. So if you take too much creatine, you put a lot of stress on your kidneys in what they're doing. So they say, drink a bunch of water to help flush the kidneys from accumulating. Make sense? But the idea is this. If we increase the amount of creatine phosphate, we could potentially produce more ATP to let my muscles do a couple extra reps. And literally that's what it goes through. You go through that very quickly, right? A couple extra reps of an exercise. Okay? Questions? But immediate transfer, produce ATP quickly. Okay? Short-term glycolysis review does not require oxygen, which means it's what? What's the word? Anaerobic. Okay? Glucose, in this case, is sourced or the origin of the glucose that's used is from a muscle's storage of glucose in the form of glycogen. All right? Glycogen is then, glucose molecules are clipped off the end of glycogen, and then they go through glycolysis. Or it could come through blood glucose, depending on if the muscle is deprived of glycogen or not. Okay? Converted to 2 pyruvate molecules, we produce 2 ATP per glucose, and this occurs in the cytosol. Okay? Pretty straightforward. All right? What else is produced? What's left off here? What else is produced? We also produce 2 what? Right? 2 NADHs are left. And the NADH does not like to be in this form. It likes to dump its electron somewhere, typically in the electron transport system, and then go to NAD+, back to NAD+, and then back to there, right? It's just shuttle. Okay? If oxygen is not present, what happens? Well, let me rewind. If oxygen is present, where does this NADH dump its electron? Goes to the electron transport system, right? Electron transport chain. And we produce ATP through oxidative phosphorylation. What happens if we don't have enough oxygen in the muscles, or we can't move enough oxygen into the muscles fast enough? Then what? Fermentation. What type of fermentation? Lactic acid fermentation. Okay? So in that case, lactic acid fermentation, okay, these 2 NADHs are still produced through glycolysis. Okay? Now they can't go to the ETC because there's no oxygen. So they end up moving this electron back onto the pyruvate, and then that is broken down into produce lactic acid. Okay? And there's no excess carbon dioxide. So when we go outside and we run a bunch of wind sprints back and forth on the quad from here to the library, what happens to your muscles? How do they feel? You've all done it. Yes? You've done high intense exercise for a very short period of time. And what do you feel? Someone said it. It burns. You feel the burn, right? We all feel the burn. That burn is a buildup of lactic acid in the muscles. Because the muscles are trying to produce ATP, but your body physically can't get enough oxygen into the muscles through breathing and what you're doing, high intense exercise. You can't get enough oxygen into the muscles fast enough, so they switch to an anaerobic process to produce ATP, and you build up lactic acid. So then after that, all right, trainers will say, all right, stretch it out, move it around, clear that lactic acid from your body, right, from your muscles, so it doesn't hurt the next day, okay, or sting or whatever else. That's lactic acid fermentation. Okay, so our long-term supply comes from oxygen, right? Pyruvate is oxidized to carbon dioxide. We've gone over this, and we can break down, we use NADH and FADH, and we produce all of our ATP. Well, the bulk, we produce more ATP from cellular respiration than we do from lactic acid fermentation or glycolysis or even immediate transfer systems, okay? So the vast amount of ATP is produced in this long-term, and it's a slower process, okay? We use glucose as the ideal starting point, but triglycerides and fats can also be used as a fuel, and even proteins to produce ATP, okay? And fat is one of our energy storage molecules, right? So when we break down fat, we can produce more ATP from those longer chain triglycerides. Okay, so when we break down fat, we can produce lots and lots of ATP that are used for muscles. Okay, so here are your metabolic processes. Again, this is a review: immediate transfer, short-term, and long-term supply. Okay, questions? So, the source then, how we use our ATP, depends on the intensity and duration of exercise. Okay? I gave you an example of a 5-second flex versus wind sprints, that happen on the quad, okay? So for a 50 meter, less than 10 seconds, right? Quick duration, very quick, and done. Phosphate transfer system. Okay, we'll produce ATP by just taking what's already there, shuffling it around to produce as much ATP as possible that they're used. Okay, cool. For a longer exercise, such as a 400 meter sprint, less than a minute, still high intensity, sprinting. Okay, ATP is supplied primarily by glycolysis, right, after the first few seconds. So 5 seconds of transfer, and then glycolysis from there on out. After a 400 meter sprint, the Olympics just stopped. What did those athletes look like? Probably the best trained athletes in the world. What do they look like? At the finish line, what do they look like? Give me an example of why you know they're tired. They're breathing. How? Heavily. Okay? Right? Some of them are bent over. Okay? All right. Why are they breathing so heavy? Well, here's what I'm asking. Is they just did an exercise that basically did not require oxygen. So why are they breathing heavy? Think about that. Hold that in your mind for a second. Okay? Let's talk about the 1500 meter run. Not necessarily a sprint, but run. Little less intensity. Okay? But it's longer duration. Over that time, an endurance athlete, for instance, okay, still run at a high speed, but it's a longer period of time and a little less intensity. Most of the ATP in this case comes from cellular respiration. Okay? What's the difference? Difference in intensity, right? And because there's a difference in intensity, the endurance athlete, the longer period, can deliver oxygen to the muscles adequately to produce cellular respiration. The respiratory system and the blood are moving oxygen into the muscles at a high enough rate to supply it to produce and allow oxygen for cellular respiration to occur. Make sense? In the 400 meter sprint, you're not doing that. Higher intensity, shorter duration, right? So in the sprint, you run out of oxygen in the muscles, okay? And now, at the end of that sprint, you're breathing heavily because you owe the muscles some oxygen. Some of the muscles store oxygen. The muscles use that in that sprint. They use their storage. They're coming to collect what you used. And you have to return that, right? It's what we call an oxygen debt. All right, so here's kind of the energy storage and kind of how long it happens, right? High intense exercise utilizes ATP quicker, right? Lower intensity endurance shifts over to cellular respiration. That's why we have that fat burning zone when you're doing aerobic exercises, right? That allows the breakdown of fat, utilize glucose, and utilize cellular respiration for delivering ATP. So now that oxygen debt, okay, is the amount of oxygen you used during an exercise, and now you have to return back up to that pre-exercise condition. You have to return what you used. So you have to return that through respiration, through breathing, right? Oxygen loading onto hemoglobin through the body and delivering that to muscles. On top of that, you have to then reload hemoglobin, reload myoglobin, replenish any glycogen that is present in the muscles, restore ATP and creatine phosphate, and then also, by the way, get rid of that additional lactic acid and convert that to glucose that can be used as well. Okay? So after exercise, there's still a lot of stuff that has to happen, what we call, quote unquote, our recovery period, right? Why do you have a recovery drink after a workout? Look to see what's in it, right? Read the ingredients. There's some stuff in there that's going to help these things to happen. Make sense? Yes? Right? Yes? So oxygen jet, ATP is related, or oxygen and how it's used is related to how ATP is used during exercise. Okay? And how we can supply ATP to muscle. Okay? In order for contraction to occur. Okay? Any questions?